Isn't it amazing, though, how a tiny molecule, a drug, can orchestrate such huge changes inside us? It's like this miniature conductor leading a biological orchestra.
It really is fascinating.
And today on the deep dive, we're really getting into the nuts and bolts of pharmacodynamics. How drugs actually work down at the molecular level.
Yeah, and for you listening, if you want to get up to speed on this quickly, this is kind of your shortcut. We've waded through the dense science to pull out the core ideas of how medicines actually produce their effects without you getting bogged down in every single detail.
Exactly. Our source today is a pretty comprehensive chapter covering pharmacodynamics. It hits everything from the receptors drugs target to the signaling inside cells, even how new drugs get designed.
It's a journey right into the fundamentals of how medicines do what they do.
So our mission, unpack the key principles of how these drugs interact with our body's machinery to actually help us. We want you to have that aha moment.
Without the headache of a textbook.
Hopefully. Okay, let's jump in. Pharmacodynamics. In simple terms, what are we talking about?
Okay, so pharmacodynamics is essentially the study of what drugs do to the body, and importantly, how they do it. We're looking at the biochemical, the cellular, the physiological effects.
The mechanism of action?
Exactly, the mechanism of action. It's not just knowing flipping the switch turns on the light, it's understanding the wiring, the filament, the whole circuit.
And a big part of that circuit involves these drug receptors or targets. They sound pretty central.
Oh, absolutely central. They're usually proteins, macromolecules in our cells that drugs bind to. That binding is what kicks off a response. But here's a key thing. Drugs usually modify biological processes that are already happening. They don't typically invent completely new functions for a cell. They tweak the existing system.
Okay, so they're tweaking the system. Where do we find these receptor targets? Are they all just sitting on the cell surface like little docking ports?
Many are, yeah. On the cell surface, receiving signals. But not all. Some are inside the cell, maybe in the nucleus, where they can actually affect gene expression.
Wow.
And some are even extracellular. Think about drugs that target clotting factors circulating in your blood. Those aren't inside a cell.
Right. The source also mentions acceptors, like serum albumin. They bind drugs too, but they're different.
Yeah, that's a good distinction. Acceptors like albumin bind drugs, sure, but they don't trigger a direct biological effect themselves. Their main job is more in pharmacokinetics.
Oh, how the drug moves around.
Exactly. Distribution, metabolism, excretion. They act like reservoirs, affecting how much free drug is available to actually reach the real receptors and do its job.
Okay, got it. Now, things get really cutting edge here. The chapter talks about therapeutic biologics, engineered antibodies, enzymes, and even, oncolytic viruses, gene therapy.
Yeah.
How do they fit in? They seem way different from a standard pill.
They represent a huge leap, definitely. Their pharmacodynamics can be fundamentally different. An oncolytic virus, for example, its mechanism is literally infecting and killing cancer cells.
Direct action.
Very direct. Gene therapy aims to fix the underlying genetic problem. Monoclonal antibodies are designed to be incredibly specific, maybe latching onto just one type of protein involved in a disease. So how it works is often much more targeted than many traditional small molecule drugs interacting with broader receptor systems.
Okay, let's circle back to those physiological receptors, the ones that normally respond to our body's own signals. The chapter uses terms like agonist, antagonist, partial agonist, inverse agonist. Can we break those down?
Absolutely. Think of a physiological receptor like a lock, and the body's natural signaling molecule, the endogenous ligand, is the key.
Okay.
An agonist is like a drug key that also fits the lock and turns it. It mimics the natural key's effect, activating the receptor, turns the signal on.
So it stimulates the receptor.
Right. A primary agonist binds where the natural key does. An allosteric agonist binds somewhere else on the lock, but still helps it turn, maybe making it easier for the natural key.
Interesting.
And understanding these, especially some types of receptors, is crucial. If you can design an agonist that only hits the receptor subtype you want, say, but not the lungs, you get fewer side effects. That's the goal.
Right. Selectivity. Okay, so agonists are on. Antagonists must be the off switches then.
Pretty much. Antagonists bind to the receptor, occupy the lock, but they don't turn it. They just sit there.
Blocking it.
They prevent the natural key, or an agonist drug, from binding and activating it. A syntopic antagonist competes for the same spot as the agonist key.
Okay.
An allosteric antagonist binds elsewhere but changes the lock shape so the agonist key doesn't work well. Then you have chemical antagonists that just grab the agonist molecule directly before it even reaches the receptor. And functional antagonists work through a whole separate pathway to produce an opposing effect.
Wow, lots of ways to block something.
Indeed.
And what about partial and inverse agonists? They sound a bit trickier.
They are a bit more nuanced. A partial agonist is like a key that fits and turns the lock, but only partway. No matter how many keys you put in, you never get the full on signal you'd get from a full agonist.
So a dimmer switch effect.
Kind of, yeah. It produces a submaximal effect. Now, an inverse agonist is even more interesting. Some receptors have a low level of activity even when no key is in the lock, a sort of baseline hum.
Right.
An inverse agonist binds and actually stabilizes the receptor in a truly inactive state, turning that baseline hum down. So it reduces activity below the level seen with no agonist present at all. Examples include receptors for benzodiazepines, histamine, opioids. And here's a twist. If you have a full agonist present, both partial agonists and inverse agonists can end up acting like competitive antagonists. They get in the way and reduce the maximum effect the full agonist can produce.
Okay, that makes sense. The chapter then talks about drug specificity, affinity, and intrinsic activity. How do they relate to the drug's structure?
Right, two key concepts. Affinity is simply how tightly the drug binds to the receptor. Think of it as stickiness. Measured by the dissociation, constant, lower number, tighter binding.
No. Yeah.
Intrinsic activity is what the drug does after it binds. Does it activate the receptor? How strongly? That's efficacy, essentially. A drug's chemical structure determines both how well it fits the receptor affinity and what it does once it's there. Intrinsic activity. Even tiny tweaks to the molecule can dramatically change these.
And that dictates specificity. High specificity sounds good, low specificity. Maybe not so much. You mentioned amiodarone earlier.
Exactly. High specificity means a drug mostly hits one type of receptor in specific tissues. Focused effect, usually fewer side effects. Low specificity means it can bind to multiple receptor types in different places.
Which might be useful sometime.
Sometimes, yes, for broader effects. But it definitely increases the odds of off-target effects, side effects. Amiodarone, used for heart rhythm, its structure looks a bit like thyroid hormone, so guess what? It can mess with thyroid receptors too.
Ah, okay. And what about racemic mixtures and stereoisomers? I kind of assumed one drug molecule was like any other.
A very common thought. But many drugs are mixtures of stereoisomers molecules that are mirror images, like your left and right hands, chemically similar, but their 3D shape is different.
Right.
And that shape difference can mean they bind to receptors differently, or the body handles them differently. Sotalol for the heart, one hand is great at blocking potassium channels. The other hand is also a good beta blocker. So they have different pharmacodynamic and pharmacokinetic properties.
So one drug might act differently depending on the receptor, the tissue, the dose, even the person?
Precisely. A drug might hit receptor A at low doses, but start hitting receptor B at higher doses. Its ability to get into the brain versus stay in the blood matters. And pharmacogenetics, individual genetic differences in receptors, plays a huge role too. Plus, interactions with other drugs, it gets complex fast.
And our bodies adapt, right? Downregulation, desensitization, tachyphylaxis. It sounds like receptors can get tired of being stimulated. Is that why some drugs seem to lose effectiveness over time?
That's a major reason, yes. The body strives for balance. Continuous agonist stimulation can lead to downregulation. The cell actually reduces the number of receptors on its surface.
Fewer targets.
Fewer targets. Desensitization is often faster. The receptor is still there, but it becomes less responsive to the agonist binding. Tachyphylaxis is super rapid tolerance. Effect diminishes very quickly, like with nitroglycerin sometimes. The body is essentially saying, okay, too much signal, let's dial it back.
And the flip side is drug resistance, like antibiotic resistance.
Exactly. Resistance can happen in a few ways. Pharmacokinetic resistance is when the body gets better at eliminating the drug, so less reaches the target. Or pharmacodynamically, the receptor itself can change, maybe through mutations, so the drug just doesn't bind or activate it properly anymore. Huge issue in cancer therapy and infectious diseases.
But not all drugs need these complex receptors, right? Antacids, mannitol, they work differently.
Correct. Some work through simpler mechanisms. Antacids just chemically neutralize stomach acid. No receptor binding needed. Mannitol works by osmosis, drawing water out. And anti-infectives often target structures or processes that are unique to the microbe, not present in us humans. That's their form of specificity.
Let's talk about drug design and structure-activity relationships. It sounds like chemists are molecular architects, tweaking structures.
That's a great way to put it. Understanding how a drug's shape and chemical properties relate to its biological activity, the structure-activity relationship, or SAR, is fundamental. Even if we don't know the exact receptor for some drugs we call those orphan receptors, we can still learn from SAR.
So,
By making systematic changes to a known drug molecule, creating related compounds called congeners, we can see how those changes affect affinity, efficacy, or even how the body handles the drug. The goal is often to improve the therapeutic effect while reducing side effects, or make it target one receptor subtype over another.
and technology helps massively here, I assume. Computers, modeling.
Oh, hugely. Computer analysis helps identify the pharmacophore, the key features of a molecule needed for activity. Molecular modeling lets us visualize potential drug-receptor interactions. Knowing the actual 3D structure of a receptor, say from X-ray crystallography, is like having the blueprint for the lock, invaluable for designing the perfect key.
And pharmacogenetics you mentioned, knowing how individual variations affect things.
Yes, that's adding another layer. Understanding how genetic variations in receptors or metabolic enzymes influence drug response allows for much more tailored, personalized medicine down the line.
Okay, let's get quantitative. Receptor occupancy theory, dose-response curves. These sound basic, but crucial.
They are foundational. Receptor occupancy theory, stemming from the law of mass action, basically says the drug effect is related to how many receptors are occupied. The dose-response curve visually shows this relationship.
S-shaped curve usually.
Often, yes, especially on a semi-log plot. It shows us the minimum dose for an effect threshold, how quickly the effect increases with dose slope, and the maximum possible effect, maximal asymptote, or Emax.
But sometimes you get weird shapes, like U-shaped curves with hormesis. That sounds counterintuitive.
It can be. Hormesis is where low doses give one effect, maybe beneficial, and high doses give the opposite, often toxic effect. Seen with some prostaglandins, neurotransmitters, it reminds us that the dose makes the poison or the cure, and more isn't always better.
And clarifying affinity, efficacy, potency, people mix these up. So affinity is stickiness.
Right, how strongly it binds.
Efficacy is the maximum effect it can produce once bound.
Correct, how big the response can get, Emax.
And potency is about how much drug you need to get a certain effect, often half the max effect, the EC50.
Exactly. A more potent drug gives you that effect at a lower concentration. Potency is about the dose needed. Efficacy is about the maximum response achievable. They are distinct. A very potent drug might not actually be very efficacious, and vice versa.
Got it. And when antagonists block agonists, there are different flavors of that too.
Yes. Competitive antagonism is reversible. The antagonist and agonist compete for the same binding site. If you add enough agonist, you can overcome the block and still reach the maximum effect. It just shifts the dose-response curve to the right. You need more agonist.
Okay, so it's surmountable.
Right. But with irreversible or pseudo-irreversible antagonism, the antagonist binds so tightly, maybe covalently, or dissociates so slowly that it effectively removes receptors from the pool.
Like taking chairs out of the room permanently.
Exactly. So even with tons of agonist, you can't reach the original maximum effect. The Emax goes down, and the curve shifts right too.
An allosteric antagonist binds somewhere else entirely.
Correct. They bind to a different site on the receptor, changing its shape. This might reduce the agonist's affinity or its ability to activate the receptor once bound, or both. It can lower the Emax and/or shift the curve right. And remember, allosteric sites can also be targets for drugs that enhance agonist effects, allosteric potentiators or agonists.
Right. Now, combining drugs, additivity and synergism. Sometimes 1 + 1 equals 2, sometimes 1 + 1 equals 3.
That's the idea. Additive means the combined effect is just the sum of the parts. Synergistic or superadditive means the combination gives a bigger effect than you'd expect just by adding them up.
Which could be good clinically. Lower doses maybe.
Exactly. Lower doses of each, potentially fewer side effects. The opposite is subadditive or negative synergism, where the combined effect is less than the sum. We can visualize these interactions using isobolograms, comparing the doses needed for a certain effect alone versus in combination.
It's clear there's huge variability in how people respond. Definitely not one size fits all.
Not at all. Age, disease state, genetics, other drugs, they all play a role. Receptors themselves change over time. We analyze this population variability using quantile concentration effect curves.
Looking at the fraction of people responded.
Yes, that gives us the ED50, the dose effective in 50% of the population. And in preclinical studies, the LD50, the lethal dose in 50% of animals.
Which leads to the therapeutic index. Safety margin, basically.
Right. The ratio LD50, ED50 gives a rough idea of safety. A bigger index is generally better. The therapeutic window is maybe more clinically relevant, the range of concentrations that gives efficacy with minimal toxicity in most people.
But still need to monitor individuals.
Yeah, absolutely. These are population averages. Individual responses can vary widely. Figure 3 to 8 in the source lists a ton of modifying factors, errors, compliance, pharmacokinetics, genetics, interactions, tolerance, placebo effect. It's a long list.
And drug interactions can be pharmacokinetic, affecting absorption, metabolism, etc., or pharmacodynamic, right, where the drugs interact at the action level. Examples.
Classic pharmacodynamic interaction. Nitrovasodilators like nitroglycerin boost CGMP levels. PDE5 inhibitors like sildenafil block CGMP breakdown. Take them together.
Too much CGMP effect. Blood pressure plummets.
Dangerously low. Another big one is warfarin, the blood thinner. Interacts pharmacodynamically with vitamin K, antibiotics affecting gut flora, NSAIDs, aspirin all affecting clotting differently, increasing bleeding risk. And we have to be extra careful with kids and the elderly due to changes in both pharmacokinetics and dynamics. The Beers criteria helps guide prescribing for older adults.
Okay, let's shift to the specific mechanisms. How do drugs actually do these things? First up, changing concentrations of our own signaling molecules.
Yeah, a very common strategy. Interfere with the life cycle of neurotransmitters or hormones. Block synthesis, block storage, block release, block reuptake from the synapse, block breakdown.
Like SSRIs blocking serotonin reuptake.
Exactly. Or drugs affecting adrenaline pathways, acetylcholine, dopamine. Same principles apply to peptide hormones like ACE inhibitors affecting angiotensin, or NSAIDs blocking prostaglandin synthesis via cyclooxygenase inhibition.
Then there are drugs acting outside cells, targeting enzymes or molecules involved in things like clotting or inflammation.
Right, targeting coagulation factors in the blood, or enzymes driving inflammation in the extracellular space, not binding to a typical cell surface receptor.
And anti-infectives targeting microbial-specific pathways. The CRISPR idea for malaria in mosquitoes is wild.
Isn't it? Using gene editing to make the vector resistant to the parasite. A totally different way of thinking about disease control, based on understanding those molecular interactions.
Some drugs also mess with ion balance pumps and transporters, especially in the kidney and gut, like diuretics.
Precisely. Diuretics target specific ion transport systems in the kidney tubules to make you excrete more salt and water. Proton pump inhibitors block the acid pump, H plus K plus ATPase, in the stomach lining. Direct action on those transport proteins.
Okay, now the really deep dive. Intracellular signal transduction pathways. How the signal gets from the receptor across the membrane and causes changes inside the cell. Sounds complex.
It can be, but the core idea is a relay race. Ligand binds receptor, that's a start. The receptor then activates molecules inside the cell, often via intermediary transducers.
Like G proteins.
Like G proteins, exactly. These transducers then influence effectors, enzymes, or ion channels. These effectors often generate small, mobile second messengers like SAMP, calcium ions, IP3, nitric oxide. These little messengers then spread the signal further inside the cell.
And it's not just random diffusion, right? Compartmentation, scaffolding proteins, it's organized.
Highly organized. We know signaling components are often clustered together in specific microdomains within the cell, held by scaffolding proteins like AKKs. This allows for speed, efficiency, and specificity. Keeps the signal localized where it needs to be.
Like little signaling hubs.
Ah.
And cells integrate multiple signals.
Absolutely. A single cell receives many inputs. The signaling networks integrate these, often involving amplification steps where one activated molecule can trigger many downstream molecules. It allows for complex, fine-tuned responses.
Let's talk receptor families. Table 3.1 lists them. G-protein-coupled receptors, UPCRs, are huge.
The biggest family by far. Target for, maybe a third or more of all drugs? They have that classic 7 transmembrane helix structure. Bind an incredible variety of things, neurotransmitters, hormones, light, smells, involved in almost everything.
And GPCR subtypes, like beta-1 versus beta-2 adrenergic receptors, are key for drug targeting.
Crucial. Allows for selectivity. Designing a drug that hits beta-1 in the heart without hitting beta-2 in the lungs avoids unwanted side effects like bronchoconstriction. Exploiting subtype differences is a major goal in drug development.
What about GPCRs forming dimers, pairing up? Functional significance.
Yes, increasingly recognized. GPCRs can form pairs, homodimers or heterodimers, or even larger complexes. This dimerization can affect ligand binding, G-protein coupling, signaling duration, how the receptor is internalized. It adds another layer of regulation and potential drug targeting.
And the G proteins themselves, the link between receptor and effector.
Right, they're heterotrimers, alpha, beta, gamma subunits. The alpha subunit binds GTP or GDP. When the receptor is activated, it swaps GDP for GTP on the alpha subunit.
Which activates the G protein.
Exactly. The GTP-bound alpha subunit, and sometimes the beta-gamma pair, then interacts with downstream effectors like adenyl cyclase or phospholipase C. There are different families of G-alpha proteins, Gs, G, GHU, G1213, linked to different pathways. The signal stops when GTP is hydrolyzed back to GDP, often helped by RGS proteins, and prolonged stimulation can lead to desensitization involving GRKs and arrestins. It's a dynamic cycle.
So GS often activates adenyl cyclase, making CAMP, the classic second messenger.
That's a major pathway. CMP then typically activates protein kinase A, pKa, but TAP can also directly affect other things like certain ion channels or EPEC proteins, and CMP levels are controlled by phosphodiesterase, PDEs, which break it down.
And pKa then phosphorylates other proteins.
Yes, pKa adds phosphate groups to specific serine or threonine residues on target proteins, changing their activity. This propagates the signal. And AKPs, those anchoring proteins, help ensure pKa acts on the right targets in the right place.
There's also cGMP, acting similarly via protein kinase G. PKG.
Correct. cGMP is generated by guanylyl glycolases and broken down by PDEs. It activates PTG, which phosphorylates its own set of targets, very important in smooth muscle relaxation, vasodilation.
And PDEs are drug targets themselves.
Yeah.
Right? For asthma, erectile dysfunction.
Absolutely. Blocking specific PDEs, like PDE5 inhibitors for ED, increases levels of CAMP or cGMP in specific tissues by preventing their breakdown, enhancing those signaling pathways.
What about EPACs, another CAMP target besides pKa?
Right. EPACs are guanine nucleotide exchange factors activated by CAMP. They activate small GT passes called RAP. They mediate some CAMP effects independently of pKa, involved in things like insulin secretion.
Okay, another big pathway. GQ activating phospholipase C, PLC, leading to DAG, IP3, and calcium release.
Yes, another major route. GQ activation leads to PLC cleaving PIP2 in the membrane. This yields DAG, which stays in the membrane and activates protein kinase C, PKC, and IP3, which diffuses and binds to receptors on the endoplasmic reticulum,
triggering calcium release from internal stores.
Exactly. That spike in intracellular calcium is a powerful signal. Calcium itself acts as a second messenger, activating calcium-dependent enzymes, including PKC isoforms and comodulin-dependent kinases. Lots of downstream effects.
Moving to ion channels. Crucial for electrical signaling.
Fundamental. They allow ions to cross membranes, creating electrical currents. Essential for nerves, muscles, heart. Their open and closing is tightly controlled.
By voltage changes or by ligands binding?
Both. Voltage-gated channels for sodium, potassium, calcium chloride open or close in response to changes in membrane potential. They drive action potentials. Think local anesthetics blocking sodium channels, or calcium channel blockers used for hypertension. Ligand-gated channels open when a specific molecule, like a neurotransmitter, binds directly to the channel protein, key for synaptic transmission.
Like the nicotinic acetylcholine receptor at the neuromuscular junction.
The classic example. Acetylcholine binds, the channel opens, ions flow, muscle contracts. Slight differences between muscle and neuronal nicotinic receptors allow drugs like rocuronium to selectively block the muscle ones during surgery. Other key ligand-gated channels respond to glutamate, GABA, glycine in the brain,
and TRP channels.
Yeah.
Involved in sensing things.
Yes. Transient receptor potential channels. A diverse family involved in sensing temperature, pain, pressure, chemicals. Involved in many sensory functions. They're becoming interesting drug targets for pain and inflammation.
Okay, next category. Transmembrane receptors linked to intracellular enzymes. Receptor tyrosine kinases, or TKs first. Growth factors, insulin.
Very important. These receptors have kinase activity built into their intracellular domain. Ligand binding usually causes them to dimerize, activating the kinase. They phosphorylate themselves and other proteins on tyrosine residues,
creating docking sites.
Exactly. Docking sites for other signaling proteins containing SH2 domains. This recruits signaling complexes and activates downstream pathways like the RAS-MAPP pathway, crucial for cell growth, and the PI3K-ACT pathway, important for cell survival. Dysregulation here is common in cancer, making RTKs major oncology targets.
And the JAK-STAT pathway for cytokines, different mechanism.
Slightly different. The cytokine receptors themselves don't have kinase activity. They associate with separate intracellular tyrosine kinases called JAKs. Ligand binding brings the receptors and JAKs together. JAKs get activated, phosphorylate the receptor, and then phosphorylate STAT proteins,
which then go to the nucleus.
Right. Phosphorylated STATs dimerize, move to the nucleus, and regulate gene transcription for immune responses, inflammation, etc.
Receptor serine threonine kinases, like RTKs, but different amino acids.
Exactly. Activated by TGF-beta family ligands. They phosphorylate serine and threonine residues on downstream SMAD proteins, which then regulate gene expression. Important in development, wound healing.
Toll-like receptors, TLRs, for innate immunity.
Yes, they recognize pathogen patterns. Ligand binding triggers a cascade involving adapter proteins like MyD88, ultimately activating NF-kappa B and inflammatory gene expression. No intrinsic enzyme activity.
And TNF-alpha receptors, also activating NF-kappa.
Yes, via a distinct pathway involving recruitment of proteins like TRADD and RAP kinases. Leads to inflammation. That's why anti-TNF therapies like infliximab are effective in some inflammatory diseases. They block the initial signal.
Some receptors directly make cGMP, natriuretic peptides, nitric oxide.
Correct. Natriuretic peptide receptors are guanylate cyclases. Ligand binding activates the enzyme domain inside the cell. Nitric oxide, NO, produced by NO synthase, diffuses into cells and activates a soluble guanylate cyclase, SGC, in the cytoplasm. Both pathways increase cGMP, leading to vasodilation via PKG.
Nuclear hormone receptors are different again. They go right to the DNA.
They do. They bind lipid-soluble ligands like steroids, thyroid hormone, vitamin D. The ligand receptor complex then binds directly to specific DNA sequences, HREs, and recruits co-activators or core-pressors to modulate gene transcription. Direct link between signal and gene expression. Hugely important in metabolism, development, reproduction.
And finally, apoptosis and autophagy pathways, cell death and recycling.
Crucial for tissue health. Apoptosis is programmed cell death, essential for removing old or damaged cells cleanly. Involves caspase enzymes. Can be triggered externally, death receptors, or internally, mitochondrial stress. Defects are linked to cancer. Autophagy is the cell's cleanup crew, degrading damaged components and recycling materials, important for surviving stress. Dysfunctional autophagy is implicated in neurodegeneration and other diseases. Both are complex signaling networks and potential drug targets.
And looking ahead, pharmacotherapies that directly target genes, antisense, RNAi, CRISPR.
That's the cutting edge. Antisense oligonucleotides, ASOs, block mRNA translation. RNA interference, RNAi, uses small RNAs to silence genes. CRISPR allows precise DNA editing. These offer revolutionary ways to treat genetic diseases by correcting or compensating for the underlying defect at the source.
The final example, the arterial wall, integrating all these signals, contraction, relaxation, really ties it together.
It's a perfect illustration. Smooth muscle, endothelium, nerves, all communicating via different receptors and pathways, GPCRs, ion channels, enzyme-linked receptors, responding to various signals, neurotransmitters, hormones, local factors like NO, to control vessel tone.
Angiotensin II, norepinephrine causing contraction via GQ-PLC calcium.
While NO and natriuretic peptides cause relaxation via cGMP and PKG.
It shows why treating something like hypertension might involve targeting different points. Beta blockers, ACE inhibitors, calcium channel blockers.
Exactly. Understanding the interplay of these pathways helps us choose the right therapeutic strategy for an individual patient, targeting the specific mechanisms driving their condition.
Okay, let's wrap this deep dive. Key takeaways.
I think the main point is that drugs work by interacting, usually very specifically, with molecular targets, often receptors, to modulate the body's own intricate signaling pathways. It's about hijacking or adjusting existing communication networks.
And the complexity is just astounding, but hopefully you, our listener, now feel you have a better grasp of that fundamental language, not just what a drug does, but how it achieves its effect at that molecular level.
It really is empowering knowledge. And maybe a final thought to leave you with. Consider how this ever-deepening understanding of molecular mechanisms is driving personalized medicine. Imagine treatments tailored not just to the disease, but to your specific version of it, based on your unique genetic makeup and the precise molecular pathways involved. The potential for more targeted, more effective therapies is just immense as we learn more.